Hi everyone, in this video I'll be talking about mantle cell lymphoma and marginal zone lymphoma. This is the video of my lymphoma and leukemia series. And mantle cell lymphoma is a very rare non-Hodgkin lymphoma which occurs mostly in males. It has a preponderance for males. And what is the origin? Then it arises from the mantle zone B cell. Now the origin is from the mantle zone B cell, and I've already told you that mantle zone B cells are those B cells which are you can say which have been selected for antigen presentation, since they are not presenting a self antigen, they are presenting a proper antigen. Now what is the pathogenesis? What happens is that there is chromosome 11-14 translocation which causes the cyclin D1 gene on chromosome 11 to translocate alongside the immunoglobulin gene present obviously on the chromosome 14, and this causes the enhanced transcription of you can say cyclin D1 gene. Now basically these mutations or this type of lymphoma is very rare because mantle B cells are not that kind of cells which are very prone for mutations, but yes, mutations can happen anywhere, so this happens. And also mantle B cells are the only cells which transition and then become centroblasts, and the centroblasts are obviously the cells which are involved in, you can say, the VDJ recombination process in which we produce the perfect antibody. Now what are the clinical features? The clinical features is that the patient presents with a painless lymphadenopathy, that lymph nodes are enlarged and swollen, and also there is an increased presentation in extranodal sites like the spleen, liver and the gut also. Now you will see that the patient will present hepatosplenomegaly, but why the gut? Why is it presenting in the gut? So the food that we eat is also a kind of an antigen, and in our gut there is mucosa associated lymphoid tissue. You must have heard about it. So the mucosa associated lymphoid tissue consists of lymphoid follicles. And what is the function of lymphoid follicle, whether uh, it is in lymph nodes or whether it is in an extranodal site, is that it is constantly surveilling the antigens. It is constantly seeing the antigen and checking that, oh, is this antigen pathogenic or not pathogenic. So and what are the mantle B cells? What is their function? They are those B cells which have been selected for antigen presentation. And where is the antigen presentation going on very much? Obviously in the lymphoid follicles. That is why the gut is also involved, but not that commonly it is involved, but yes, occasionally it can be involved. Now if I talk about the lymph node biopsy, then this is has been taken from a normal lymph node, and as you see here that the mantle zone and the germinal center are properly demarcated here, and this is the capsule, and this whole is the lymphoid follicle. Now if I take the image, this is the image of you can say the lymph node biopsy of a patient having mantle cell lymphoma, then you can see that a dark region is effacing a very lighter region. This zone or this follicular zone is very small. See, it is so small. Here it is even not present here. It has vanished. So that is what is happening here, that when the mantle B cells, since their normal mantle B cells are involved in the differentiation to centroblast, and then these centroblasts become centrocyte. But when the mantle B cells are multiplying in an uncontrolled fashion due to the increased expression of cyclin D1, they will efface the whole lymphoid follicular architecture, and the follicular zone which is present in the center will be completely gone, or it will be very atrophic in nature. You won't see centrocyte or centroblast in that follicular zone. Now if I talk about the gross image, then you see that the spleen of a patient having mantle cell lymphoma presents with a micronodular pattern. These white micronodules, which are none other than the you can say the enlarged lymphoid follicles comprising of the mutated mantle B cells. And this also has been taken from a gut of a patient having mantle B cell lymphoma, and you can see that there are irregular outgrowths from the surface. These irregular outgrowths are called polyps. I will talk about polyps in later in the GIT chapters, but yes, these are the outgrowths or the polyps which you see. And why? Because just of the growth of the mucosa associated lymphoid tissue, they have arised, or you can say giving that outgrowth, polypoid outgrowths in the stomach. And if I talk about the cytology, then what do you see here is that you see mature B cells. See, the mantle B cells are also mature, and they are not that transcriptionally active. So they have a condensed chromatin and a cleaved nuclei. They have cleaved nuclear contours, and the cytoplasm is also scanned. So they uh, you say more or less they resemble a naive B cell. And what you see here is that the follicular or the germinal center here. So the germinal center, just like here also it is a zoomed image, and you see here that the germinal center is atrophic. It is not that. Generally what happens is that the germinal center has a very large space, but here you can see the mantle zone has occupied this much of an area, and the germinal center is atrophic. Now what is the immunohistochemistry markers? So the cell will be CD19 positive, CD20 positive. Obviously it is a B cell, but I told you about the mantle B cells, and they are CD23 negative. Why they are CD23 negative? Since they have been selected for antigen, the antigen they are presenting is not a self antigen, so they are CD23 negative and CD5 positive. But when they go into the germinal center, then they become both CD5 negative and CD23 negative, which I have talked about in my previous lectures. Now I will talk about marginal zone lymphoma, or the maltoma, or you can say the mucosa associated lymphoid tissue uh, lymphomas are also called marginal zone lymphoma. So what is the origin? The origin is a memory B cell. Now the memory B cell, this lymphoma originates, and let's see what why is that the memory B cell is playing a role in this lymphoma. So what is the pathogenesis? What happens is that the chronic inflammatory conditions, exactly the chronic inflammatory conditions like the H. pylori gastritis or the autoimmune disorders, which are also chronic inflammatory conditions. In these chronic inflammatory conditions, there is increased activation of memory B cells. What happens is that for every specific epitope of the antigen, a specific memory B cell is formed, which produces the perfect antibody. So what happens when an H. pylori antigen, or whenever an autoimmune response occurs, it occurs in response to an antigen. So for the every epitope of the antigen, specific memory B cells are you can say they are stimulated, and this produces a polyclonal response, causes repeated polyclonal response by memory B cell. And in this process only, due to the repeated proliferation, see when the memory B cell are called upon, so when they are called upon, they immediately transform into the activated B cells, which produce antibody, and they are also rapidly proliferating. So during this repeated process of inflammation, there will be also repeated cycles of proliferation, and due to these repeated cycles of proliferation, the memory B cell has chances to acquire an unknown mutation, which causes it to be dependent on the T cell growth factors to survive. Now you see that some of the a B cell arises which is dependent on the T cells. Since the T cells secrete the cytokines to activate the memory B cell to become the activated plasma cell. So what happens is that a kind of B cell arises which undergoes a symbiotic relationship with the T cell, in which it is dependent on its cytokines, and it actively grows, multiplies and multiplies. So this until this stage, the lymphoma is, you can say, easily regressible. It can be if it can be it can regress if the etiology like the H. pylori is cured, or if during the Jorgen syndrome, if we use corticosteroids, then yes, this lymphoma will regress. But if it is left, then it progresses. The B cell acquires even more mutations, which causes it to express a MALT1 protein. Now the MALT1 protein is the protein which increases the signaling of nuclear transcription factor. Now the nuclear transcription factor is the factor which causes expression of cytokines or growth factors from the B cell, and these growth factors help the B cell to multiply and proliferate. So it does not need a T cell now. It does not need the growth factors of T cell. Due to these mutations, or due to the progression, uh, it is capable of multiplying, and after this lymphoma then becomes very aggressive, and it can progress into DLBCL, or diffuse large B cell lymphoma, and metastasize to various organs and lymph nodes. Now what is the clinical features? So generally the lymphoma rarely progresses to that condition, and if the etiology is cured, the patient is cured of the lymphoma. But if the patient comes in an initial stage like this, which I was talking about, then the patient will obviously present with GIT discomfort. He or she will have H. pylori gastritis, and Due to that, it can also happen that due to peptic ulceration in the stomach, he or she can also present with epigastric pain. And in case of Jorgen syndrome is an autoimmune disorder in which the body attacks its own salivary glands. I will talk about it in my later videos, but the patient has salivary gland swelling and there is xerostomia, because since the salivary gland cells are being destroyed, the saliva is not being produced, and the patient has dry mouth. Now, if I talk about the lymph node biopsy, then if you take the biopsy of a patient from the mucosa of the stomach, then this image has been taken from the stomach mucosa, then you see that there are some cells, the stomach cells or peptic cells, and there is increased, you can say, the infiltration of these mononuclear cells or these lymphocytes, which you can see here. Lot of lymphocytes are present. So this is the presentation of maltoma. And if you see the gross image, then again, if you take the spleen of the patient, in which rarely it metastasizes to spleen because generally the etiology is cured, but if it metastasizes, then the patient presents with a micronodular kind of a growth. You see these arrows are also representing that. So the present with a white micronodular growth in the spleen. And if I talk about the stomach, then you see this is taken from a patient having maltoma, and this one also. You see the wall of the stomach has been thickened, the diameter has increased so much. Why? Because there is increase, you can say, increase expansion of the mucosal lymphoid tissues, and due to this expansion only, the mucosa has been uplifted, and this causes the thickening of the stomach wall. And if I talk about the IHC markers of this lymphoma, then they are CD27 positive. Obviously, the CD19 and CD20 will be positive, which is already known. But what is it that how can we differentiate it from other lymphomas? Then CD27, a special type of marker which is present in memory B cells, is positive, and BCL6 is also positive. So I end my video today. Uh, if you liked this video, give a thumbs up, comment on the comment section, and please subscribe. And I will see you in the next video. Till then, bye bye.